Hi, today I'll be discussing the paper Wireless Control of Cellular Function by Activation of a Novel Protein Responsive to Electromagnetic Fields. The purpose of this paper was to investigate a protein that can induce cellular changes through EMF, or electromagnetic fields. This is less invasive than other methods such as drugs, radio waves, or ultrasounds. The protein reported on is known as EPG, or electromagnetic perceptive gene, which is found in the anal fins of glass catfish K. fisheries. In this paper, the researchers implemented this into mammalian cells, neuronal cultures, and rat brain. EPG expression in mammalian cells and neurons increases calcium concentrations when excited by the EMF. This is probably because EMF induces a calcium influx in the electroreceptors. As seen in Figure 1, the fish swim away from the EMF from a neodymium rare earth magnet, emitting 23 milliteslas of magnetic stimulus for 10 seconds, 10 millimeters away from the fish. This is also seen in Figure 2, where the magnetic stimulus has a considerably higher distance the fish swam than the control, or sham stimulus. The EPG gene was then cloned using immature frog eggs, or oocytes. And then the putative genes were compared to GenBank's database and translated into a protein database to find a novel 133 amino acid protein that increased the current, which is the EPG. This is thought to be a homodimer, and bioinformatic analysis of the EPG predicts that secondary structure will have 5 beta strands with alpha helices at either terminus, contain a hydrophobic M-terminus signal peptide in a non-cytoplasmic region, as well as many conserved cysteines to help stabilize the non-cytoplasmic domain through disulfide bonds, as well as a predicted end glycosylation site at amino acid 77. Through cellular and region-specific expression of the EPG in vivo, they were able to explore more of how EMF affects the protein and how that will affect the cell as a whole. In mammalian cells, 68 out of 99 GFP cells in the HEK 293T mammalian cells showed an increase in calcium 2+ ions 13 seconds after an EMF stimulus. Both extracellular and intracellular Ca ions were released by the EPG EMF stimulation. Wireless activation of EPG was also done in the rodent model by injecting a virus into the limb motor cortex of a rat's brain. The contralateral forelimb in vivo here affects the left side of the rat's movements. In response to EMF caused much higher activity with the EPG in the left side of the rat compared to the control, relatively even activation for each. This provides potential for remote cell modulation with EMF in other cells